now for something, I guess, totally different. Really appreciate the organizers for including health and biomedical research in the meeting again this year. Really thrilled to share again a little bit what we've been doing over the past decade or so at our National Cancer Institute Center for Strategic Scientific Initiative. Just a brief sort of eye candy for everyone so that you guys know where I'm coming from perspective-wise. Our particular center over the past decade or so has tried to keep the Institute itself ahead of the scientific curve, so we get a lot of latitude and leeway on the types of research that we may be funding, and we tend to do a lot of pilot studies. As you can see with this little star here, that's where I joined, and I think all the fun sort of began, so very lucky to have participated in that. For those in the room that are not cancer biologists or oncologists by training, I tend to start with this slide just so that we get a set of terms out of the way and give you guys sort of a quick snapshot of state of the art in terms of cancer research thinking. But in general, I think we tend to throw a lot of words around: tumor, cancer, metastases, and I think the numbers really speak louder than my words. At this point, you know, most of the things that we tend to be able to treat and be able to look at are localized disease, where it's relatively easy to remove by either surgery. But once it really starts spreading or metastasizing, becoming more cancerous, that's when those numbers, the 5-year survival rate precipitously drops, and about 90% of these deaths are caused by the spread of disease, not necessarily that first primary tumor. And I think why we exist is sort of looking at this particular trend across the different disease spaces. About 1.6 million Americans this year projected will hear the words "you have cancer" for the first time. That's something I think everybody in the room probably, unfortunately, has somewhere in your family or in your life had that touched upon you. I think this discrepancy here when I first started was a little bit closer. I'm glad to see we are making a dent in looking at the overall death rate in 2013. But as always, it is something costly, and we do want to try to hopefully do what some of the other disease areas, such as heart disease, infectious disease, etc., make a bigger dent. And I think if you look across the US, it's also not just one area. I think there's definitely some spatial heterogeneity here. And I looked at actually this year's ERC Board of Directors, and I'm hopeful to see that hopefully some of you guys will be listening, because those dark areas are not good places. And what we can better do to sort of change those into lighter blue areas, hopefully, will be something we can discuss. And of course, it's not just a US problem, it is a global issue. By all estimates, really want you guys to focus on the purple areas where the percentage increase in cancer deaths since 2002 has grown by greater than 50%. So this is a problem that we will all be facing soon, if not already. And I think at this point, and we've heard there's not a better time to be in this type of science. I think since the publication of the first human genome in 2001, we have pretty much omicsed ourselves to death. Last count with President Obama's precision medicine initiative as the other gate. Just in the last decade, we are really putting out more publications than we can sort of read and chew on. And has that knowledge really yielded potentially more solutions for the patients? I think certainly yes, but could we make that process particularly more efficient? I think most of you probably have seen this yellow-blue, where the blue is the number of new molecular entities approved by the FDA versus how much R&D costs are put in. I think what we like to tend to highlight also is that of those new molecular entities, only a small percentage of those are truly new in class versus Me Too's. And on the other end of biomarkers, I think we're still averaging at that same rate, about 1.5 approvals per year. And I think it's not just an FDA approval concern, it's the fact that the more we learn, it seems like the less we know. So for us, obviously our budget is not infinite. We will not be able to solve all the problems. What we decided to do is take a crack at this 1.8 billion average per term, going from finding a target to successfully getting a drug approved. we really wanted to look at that first part and whether or not we can impact that first part to make sure that what goes into these clinical trials probably has the highest likelihood of success. So to do that, we asked the community in 2002, how would we get there? And if you could put your mindset back into 2002, I think some of the things that the community came back to us was that they wanted more standards and protocols, more real-time public release of data, build some large disciplinary teams, share failures and not just successes, and most importantly, begin building some of these teams that each member themselves were transdisciplinary trained. At that time, they were quite tall-tasked and particularly not friendly, especially in an NIH-type environment. But we felt that if we could actually do this, we really would have the potential to transform how we think about cancer discovery and diagnostics. So before I go too much into us, I think I just want to briefly touch upon the NIH as well. I'm assuming there are several of you that are familiar with us, but those that are not, there are 27 of us. And we do give out about that 30 billion mark that you've heard already, 80% of that out for grants, contracts, et cetera. Similarly for the NCI, we're hovering about that 5 billion mark. Again, a majority of what we do is to give that money out to the extramural community in the form of grants and contracts. We definitely this year have a changed institute. Our alternative medicine institute is now integrative health. And I think why I tend to think of NCI as number one, well, the numbers in fiscal 14 speak for themselves. We sort of are number one in that sense. For those of you guys that have had experience working with the Institute or the NIH, certainly you'll see perhaps NIBIB, NIGMS, in this particular area. I don't think that's the only gauge. And so with that, I think I want to first make sure that, because I tend to find there's a lot of NSF or DOE grantees here, a little bit different for us. I represent the program side of NIH. We do not give out the grants and we do not review those grants. We have a three-tiered sort of checks and balances system. And that I think probably separates us the most perhaps from some of your traditional granting institutes. And with that, our job is to produce, looks like in fiscal 14, about 685 different funding announcements. I tend to think of them in 3 bins. Your parent announcements, which is probably what we're most known for, give us your best idea. Specific program announcements that don't have a set aside. That sort of generally scopes the science, but we still, it's nothing like what our center traditionally uses, requests for applications, where we actually put out a directed ask. We do set aside funds, and then we pay for them. Just in terms of, to orient everyone a little bit there, what I mentioned before about, in terms of your best ideas, the black across the board at the NIH, the gray is the sort of scoped down version, whereas the blue is traditionally where we operate in and have me talk the most about. Our job, actually, from my perspective, is not just to get you guys into the blue, but move you from the blue to the gray to the black, because that's really where the money's at. And like I mentioned, we're about 27 different institutes. We all spend our money a little bit differently. Even though Cancer Institute and our infectious disease are both mission-driven, you can see that Dr. Fauci definitely spends a little bit more in the RFA space versus perhaps NIGMS that tends to spend a little bit more in the open solicitation area. And the other two, I think I just list up there in terms of scale. I don't think a bigger institute necessarily drives more RFAs, here shown in terms of our Genome Institute, where they also tend to spend a little bit more in that directed research space. And one of the things I wanted to share between last year and this year, I think, since strategic is our middle name, we've begun looking back a little bit at what the last 10 years of NIH has looked like. And I'm sure you guys are fielding some questions. We have definitely changed how we have done business. What used to be more of that program announcement, we have begun collating into parent announcements. And if you look at just the change between 14 and 04, we really did a lot more of that targeted, but not set aside type funding, the sort of gray PAR. So as you think about the NIH, I think it'd be great if you guys speak with us, talk a little bit with us, and we'll be able to give you a general sense of what that change has in terms of applying to the institutes. So coming back to the National Cancer Institute and the program itself in the US is about an $18 billion per year effort, split about 50% to private industry. The others, in terms of more public sector dollars, the NCI, while we drive some of the agenda, we're only about a quarter of that effort, with the remaining showing up in other federal programs such as the DoD Breast Cancer Program, as well as your nonprofit and your foundations. Specifically the NCI, for those of you guys that don't know us, Dr. Varmus is our director, Deputy Director Doug Lowy. For us, where I represent in green CSSI, we're about 3% of the budget, but our mission is to try to find and fund potentially the fringe. But we do make sure that what we fund does not just impact the 4 sort of main divisions of cancer research at our institute: prevention, treatment, biology, as well as population science. And that gives you a general sense of breakdown of what that black part predominantly occupies. We do spend, not by our design, but mostly by our community's needs in those particular sectors and areas. And again, I think just because it's this audience, if you guys are interested, we do put out a fact book. I think one of the things that I do want to highlight for this crowd is we've done our best over the past decade to try to keep these awards at a relatively flat rate, so there really is no precipitous drop, but it has been very difficult to continue to support some of these innovative proposals that are coming through our door. I think one of the things that also separates the Cancer Institute from some of our other NIH colleagues, we're the only ones that commands an FFRDC, or a national lab, that does allow us to pivot and move a lot faster. It is up at our Frederick, Fort Detrick area, Frederick National Lab for Cancer Research. Some of the projects I'll describe use that facility so that we can actually ramp up our efforts a lot faster versus using a grants program. Specifically for us, because we are given that leeway, we have traditionally been led by the principal deputy of the Institute. For those of you that have already heard, Dr. Varmus will be stepping down April 1st, and Dr. Lowy will be appointed acting director of the Institute after that time. So I think one of the things that we're really proud of being at the NCI and driving this type of organization is that we actually use the word sunset quite a bit. We do want to make sure that the programs that we put out are recompeted and open, and make sure that we are actually trying our best to stay ahead of that curve. Looking at it in a different way, so I'm a chemical engineer by training, so time and lens scale I think is a little bit easier for me to think through. This just generally gives you guys a sense of what type of science and team science versus individual science we have been supporting over these couple of years. And I think just to give you guys a quick flavor of how we think about it in that same engineering framework, I tend to use the sort of 101 what is water analogy. You can sort of answer that question by either looking at qualitative or quantitative type descriptors, or you can do what I'm sure many, many years ago people started taking measurements, generating some quantitative data, but unfortunately perhaps running into some disagreements. And I think there's enough engineers in this audience, I don't have to give you guys that punchline, but I think these 2 particular answers obviously are both correct, just depending on where you made that measurement, giving that new parameter of pressure. And I think that, in general mindset, is how we have been trying to change the biomedical research landscape with some of our programs. So how did we find our first couple of thermometers to actually figure out how to measure? In 2003, we launched our very first program, our Innovative Molecular Analysis Technology. That is not a typo. We do encourage milestone technology-driven, and actually, if you have too much biology hypothesis-driven research, we will return it without review. And companion to that, a sister program in which it would take some of these innovative initial sort of prototypes and actually put them to test in an emerging technologies arena. So if you looked at what the landscape looked like then in 2003, it's no surprise that genomics at that time was quite a bit of that, both in the innovative space as well as the emerging space, a little bit in proteomics, some nanotechnology, and a little bit of physics. And the gray is sort of obviously, no matter how much we put the word innovative in front of our announcements, we still get and fund some of the same old, same old. So that I think gives you a sense of what we were looking at before we started our major initiatives. And because the genomic technologies were so prevalent and rampant, we did start with that first arena. And at that time, these papers were coming out and everybody was claiming that they felt that one of these particular alterations is what drove the disease. They measured it with one technology and they did it on maybe 1 to 10 samples. I think one of the things that we really wanted to change, and again borrow and take a page from the engineering mindset, we decided to try a bold experiment in which we not only looked at one alteration, we tried to look at as many as possible for each particular tumor, do it as many times as possible for each individual cancer, do it for 500, not 10. And then do that for as many cancers as possible to see what holds true. And of course, make that data publicly available in real time. And as you can see, we were faced with a lot of thermometers at that time, different technologies. We didn't specifically exclude, we actually tried to include as many as possible so that we can get that comprehensive measurement. One of the things that we definitely learned right off the bat, I think those of you know this, water is not water is not water. We learned that as we were trying to get many of these primary tumor sections, one of the things that we found that depending on whether or not you went into a facility on Sunday versus Wednesday, your odds of being diagnosed positive changed. So those are some of the things that we are very careful about to make sure that when we actually put these tumor samples in, that we are trying our best to compare apples to apples. So what did we do? We did it for 3 particular diseases: brain cancers, lung cancers, ovarian cancers. And as I mentioned, we tried to include as many different genomic characterization techniques as possible, sequence techniques as possible, and data coordinating, as well as clinical information collected about each of those tumors. What does that look like? This is what it looks like in 2011. So each row here represents one patient, each column, 1 through 23 of your chromosomes. And as you can tell, you don't need a fancy biological degree. There are certain patterns, and not every single cancer is the same. And that we are also very lucky that we started the pilot with GBM. I think when we moved from GBM to ovarian, this is where we started realizing that perhaps we needed to talk to a few more engineers because it was not as clean. And this is what 2011 looked like. 2013, I've stopped making this graph because we are about to finish all 10,000 tumor samples that have come in. We really encourage, and if you're investigators, all of this data is publicly available. I generated this graph, and I'm a federal person, so if I can do it, your investigators can do it. I think we also wanted to take a step further and not just have one dimension of data. So we launched a second part in 2011 to augment this data set with protein data. I know that some of you guys may not remember, but DNA, RNA, protein is how our body tends to do this. So in 2011, we started and took a subsection of colon, breast, and ovarian tumor samples and did not use similar tumor samples to do genome proteome. We tried to use the same sample to do them both. And I think we are very happy to announce this paper came out last July, where we did look at both genomic and proteomic. And without looking at the figure, how many of you guys think that more than 10% of the genomic predictions came to reality in the protein space? 50%? 90%? I think the biologists, we're hoping for 90%. We averaged about 38%. So I think some of those genomic targets that people were generating some next generation therapies off of, they're sort of now rethinking, if only 30% of those actually are made into proteins, what does that mean? We hope that you and your community can help us think about what that may mean, and to really help you guys with that. Again, like the genomic part, the proteomic part data is open and available. We are sorely in need of these new computational tools to look at that particular data set. I think all 3 types, colon, breast, and ovarian, have been publicly available for some time now. We hope that you take advantage of that. And for the true chemical engineers in the room, if you don't believe what I'm saying, you can repeat the experiment yourself. We have made the affinity reagents relatively cheap. These are high quality antibodies that people can go and request, $35. And at the same time, we just launched an assay portal in which we gave out the exact protocol and how we actually conducted these experiments. So again, if you don't believe us, you can repeat it yourselves. And we hope that your constituents and your universities can help think through these problems with us. And for those of you that are interested in imaging, this is a little less, the data set is a little bit sparse at this point, but we are continuing to collect that additional dimension. So those particular samples that did have patient imaging data associated with it, that also is being culminated into the Cancer Imaging Archive. So now hopefully we'll have not just the measured genomics, proteomics, but also perhaps some spatial and temporal changes of these patients by imaging. So where do we go from here? It takes a lot to go from just collecting things and making an actual phase diagram. That's what we're really hoping you guys can help us think through. Basically help us do the reverse. I think we've done a good job of turning people into barcodes. We've not yet figured out how to turn barcodes back into people. So with that, I think I'm just going to finish with a couple of funding announcements that I think hopefully you guys will be interested in applying for. The very first of which, some of you guys may be familiar already. It's an experiment that we launched in 2009 with RO money. That first phase has completed. Dr. Larry Nagahara was able to join us today. He is helping, along with Sean and Nas, really engaging and bringing hopefully more physicists, mathematicians, engineers. They didn't think that we could get them in the room and get them to talk. It wasn't easy, but I think we've definitely proven that they're able to speak both sides of the languages. The next announcement for both, or the next receipt date for both, is November, so hopefully you guys have a lot of time to plan. Others include our Nanotechnology initiative. The Big Center initiative has already closed, but we still have both a smaller individual pilot project available, as well as training. I think that's one thing that we probably all agree on. Trying to make sure that the next generation of researchers are trained in a new fashion. That's coming up in April and May, so a little bit sooner. Certainly, some of you may have also heard this was one of Dr. Varmus's pet projects, the Provocative Question Initiative, in which, as we were thinking through some of this, we didn't want to just support teams coming up with interesting solutions to important but not obvious or unidentified, understudied problems. So these are individual R01s and R21s that we've been given out. The first time, at least at Cancer Institute, where the R01 required no or nominal preliminary data. For those interested in that particular initiative, we were approved for another 20 million. New questions are about to come out. If you've not met her already, Dr. Emily Greenspan is here with us as well. So she should be able to also answer any questions you guys may have of that particular effort moving forward. And last but certainly not least, the program that started in our center off, our IMAT program continues with another $11 million this year in fiscal 2015. As you can imagine, we have about 100 projects. Those 4 categories that I originally shared with you, they've really expanded over the couple of years that we've been putting this out. If you guys want to see what types of technology development, no biology type projects we're funding, we actually put up every single abstract from every award that we've ever given out, just so you guys can check whether or not we'd be interested. I'm pretty sure, unless you're already planning, the March date is a little too soon. But we have a June and a September date for you guys, and if you guys are interested, please email Tony, who's been doing a great job running the program for us. Just want to close by thanking all the people that I think really make my slides look great. We try our best to be multidisciplinary, to make sure that we support correctly the multidisciplinary efforts that are out there. If you guys want to learn more about us, it's our website, email. We do have a resources page. I highly encourage that you guys bookmark. We try to update that every Friday or so with potential announcements that fit people that we tend to fund. And I'm just going to leave this here for those of you guys that have junior investigators that want to learn more about NIH review. Chief Dr. Peterson, I think, gives this out and asks me to talk about it all the time. If they want to be an early career review, please send their CV to him. He'll try his best to get you guys in there. He is the overall chief of our engineering, sciences and technology study section, so been very helpful in getting folks oriented to how we do things. And I think the bottom is just their new videos about mock reviews. And then you can sort of actually see what happens behind the scenes. So thank you very much.